We usually divide the natural world into two neat categories. Animals have brains, nerves, and muscles to react in an instant and learn from their environment. Plants, on the other hand, are passive. They live in slow motion, reacting to the world over days or weeks. But there is a glaring exception to that rule. This is the Mimosa pudica, commonly known as the touch-me-not plant. If you reach out and brush a finger against its delicate leaflets, it reacts in just 0.1 seconds. Charles Darwin was fascinated by this rapid movement and studied it extensively back in 1880. Yet the exact mechanics of how a plant could move so quickly remained a mystery for over a century. What modern researchers have discovered is that the mimosa operates using a hidden internal network, mimicking an animal's nervous system, challenging strict biological definitions of how organisms learn and interact with their surroundings. When the plant is touched or wounded, it fires off a long-distance electrical impulse. This is remarkably similar to an action potential in an animal's nerve cell. In 2022, a team led by Dr. Masatsugu Toyota successfully filmed these signals in real time. They observed brilliant bursts of fluorescence, tracking the calcium ions as they raced through the plant's vascular tissue. This electrical wave travels straight to a specialized motor organ at the base of the leaf, called the pulvinus. Once the signal arrives, it forces potassium and chloride ions to rapidly flush out of specific cells inside that joint. Through a basic rule of chemistry, water follows those ions. Through osmosis, water rushes out of the cells, which immediately drops the turgor pressure, the internal water force keeping the cell rigid. The cell instantly goes flaccid and collapses. Lacking muscle fibers entirely, the plant has evolved a highly pressurized hydraulic system to snap its leaves out of harm's way in a perfect brainless reflex. Performing this mechanical feat costs the mimosa a massive amount of energy. Plus, folded leaves catch less sunlight, severely restricting the plant's ability to photosynthesize. To figure out why a plant would accept those severe penalties, Dr. Toyota's team used CRISPR genome editing to create an immotile mutant. They engineered a version of the plant lacking a pulvinus, completely unable to fold its leaves. When exposed to herbivores, the results were clear. Grasshoppers absolutely devoured the mutated, immobile plants, while largely leaving the wild, snapping plants alone. The sudden motion provides a multi-layered defense. It startles skating insects, exposes sharp thorns along the stem, and leaves the shriveled plant looking dead and unappetizing. The shrinking leaves are a highly evolved survival strategy against predators. This defense system creates an obvious problem. If the plant blindly recoils at every single physical impact, a heavy rainstorm would force it to stay closed all day, starving it of the sunlight it needs to survive. Biologists Monica Gagliano and Stefano Mancuso tested how the plant handles harmless physical contact. They built a custom rig designed to repeatedly drop water directly onto the mimosa leaves. At first, the direct physical impact of the water hitting the leaves caused them to instantly snap shut. After a few repeated drops, the plant stopped closing. It recognized that the water caused no actual damage and ceased reacting to the false alarm. Biologists call this behavioral plasticity. It is the ability of an organism to adapt its future behavior based on past experiences. The most surprising data point came later. The researchers left the plants alone, altered their lighting environment, and tested them weeks later. The plants still remembered the lesson, ignoring the water drops entirely. While the precise cellular mechanics are still being mapped out, scientists hypothesize the plant's calcium-based signaling network acts as a rudimentary chemical memory bank. The Mimosa pudica demonstrates that rapid reflexes and behavioral adaptation can exist without a nervous system. It suggests that complex responses to the environment don't necessarily depend on a central brain, but can emerge from the way a plant processes its own internal chemistry.